My name is Elayne Benson. With the help of Dr. Scholl, this past 2 semesters, I conducted a metabolic array showing basal-like breast cancer cells' ability to differentially metabolize sugar alcohols. To start, I would like to discuss a little bit about the different types of breast cancer studied here. So there are 2 main types, being luminal and basal. Luminal breast cancer is what is known to be your best prognosis. These are estrogen and progesterone receptor positive, as well as HER2 gene positive breast cancers. Basal breast cancers are more aggressive in nature. They do not contain these estrogen receptor, progesterone receptor, or HER2 positive gene receptors, which limits targeted therapeutic options, deeming it more difficult to treat. So because of this, they have higher rates of metastasis and relapse. So this differs from luminal breast cancers because luminal breast cancers are primarily differentiated. So in the upper right of the slide, you can see 2 graphs that demonstrate the rates of breast cancer relapse and metastasis in them. There is generally a high level of no relapse or metastasis in breast cancer. However, those yes lines you can see are mainly known to be those basal breast cancer cases, which do not have the ability to necessarily be therapeutically targeted for those receptors, which they do not contain. So one way to address breast cancer treatments is to deregulate the cellular energies, i.e. their metabolism. So in order to do so, the sources that increase the cell's energy must be determined, which is where we focused. So this graphic shows the difference between a normal non-cancerous cell's basic metabolic pathway on the left compared to that of a cancer cell on the right. So as you can see, it becomes more complex as it goes from glucose to the TCA cycle. So with these various detours, if you will, there are more sources of energy for metabolism to occur. So we looked at the metabolism of luminal and basal-like breast cancers under various carbon sources to determine whether these 2 metabolize differently, which leads me to the thesis of the research. So aggressive basal-like breast cancer cells have a different metabolic pattern when compared to less aggressive luminal breast cancer cells. So since we're comparing aggressive and less aggressive types of breast cancer, luminal breast cancer cells were used as a general control. MCF7 cell lines were used to model the luminal breast cancer because they are ER and PR positive with a low metastatic potential. Basal-like breast cancer was modeled with SUM159 cells. So SUM159 is a triple negative breast cancer cell line, meaning that it does not have ER, PR, or HER2 positive receptors, and it is very highly aggressive. So each of these cell lines were cultured and placed into 96 wells plates that had each well containing different carbon source metabolites. They were incubated for 24 hours before adding residurin dye for another hour, and then we read the optical density to determine the various levels of metabolism for each cell line and carbon source. So from that data, we calculated a metabolic fold. So the results showed that there was a difference in what basal-like and luminal breast cancers metabolize. So on the left is that calculated metabolic fold comparing the 2 cell lines data. So the blue represents carbon sources with highly metabolized luminal breast cancer, your MCF7s, data compared to the basal-like breast cancer, the SUM159s, while the red is comparing the highly metabolized basal-like breast cancers to the luminal breast cancers. So there were 13 metabolites that showed high levels of metabolism for luminal breast cancers compared to the basal-like, and there were 11 metabolites for basal-like breast cancer compared to luminal cell lines. So these figures show the specific types of highly metabolized carbon sources for the 11 basal-like and 13 luminal cell lines separately. So one of the most interesting findings was that the basal-like cells, uracil 159s, showed high levels of metabolism in the presence of sugar alcohol metabolites, which are these starred bars, your dextrin, lactulose, and mesoerythritol. So this is unusual because human cells do not metabolize sugar alcohols, which then poses the question of why and how human cells are unable to use sugar alcohols as an energy source, but basal-like breast cancer cells, your triple negative, are able to, and at large amounts of that, as you can see. So these are the chemical structures of the sugar alcohols that showed high metabolism for the basal-like breast cancer cells. So we have dextrin, lactulose, and mesoerythritol. All of these have alcohol groups that make them unable to naturally be metabolized by human cells, which sparks the interest of these findings. So in conclusion, the MCF7 representing luminal breast cancer cells and the SUM159 representing basal-like breast cancer cells do indeed differentially metabolize different carbon sources. It's important to note the finding that basal-like breast cancer cells showed to have the ability to differentially metabolize sugar alcohols, and because of this finding, further studies are warranted to determine whether sugar alcohols can stimulate metabolic growth in basal-like breast cancer cells. I would like to thank Dr. Scholl for helping me with this project and Paris Rizzo for giving me assistance in the lab. I would also like to thank Dr. Korkaya and Dr. Otsunova for collaboration, SE Embry for funding, and finally family and friends for supporting me. Thank you.